welcome to my channel Biopulse. We are discussing the unit of life, that is the cell. This is my fourth lecture. Welcome you all. For today's session we are going to discuss about the very important cell organelles. Okay, and this class gonna be a damn amazing because much more NEET exams questions were covered in this class, because very important organelles we are going to discuss for today. So let's get started. First, let's talk about what actually the endomembrane system. Endomembrane system is nothing but the membranous organelles is distinct in terms of its structure and functions. Whatever the cell is having their organelles, no, they have their own structure and they have their own functions, but together as an endomembrane system because their functions are coordinated. If their functions are coordinated in among them, that is considered to be a endomembrane system. What the endomembrane systems are included, which organelles are included, please keep in mind. They may ask direct question like this. Endomembrane system includes ER, that is endoplasmic reticulum, Golgi complex, lysosome and vacuoles. Okay, now endoplasmic reticulum, Golgi complex, lysosome and then vacuole. 4 endomembrane systems are included. The functions of mitochondria, chloroplast and peroxisomes are not coordinated, so these are not considered as part of the endomembrane system. Mitochondria, chloroplast and peroxisome, these also cell organelles only, but they are not coordinated with these. Okay, now that's what endomembrane system includes, only ER, Golgi, lysosome, vacuole. Why such so? For this question you will get the answer in upcoming slides. Okay, why we are going to call them as a endomembrane system only, these all things going to be clear today. Okay, now endoplasmic reticulum, this is the first organelle which is of the endomembrane system, the endoplasmic reticulum. The presence of network or reticulum of a tiny tubular structure scattered in a cytoplasm, that is called as what? Endoplasmic reticulum. See, this is the nucleus. Okay, from the nucleus what happens, there will be a network of tubules. These are the tubules structures, okay, scattered. Just imagine this is a cell, there is a cell, inside the cell what happens, there is a nucleus and inside the cell this endoplasmic reticulum is scattered. Okay, now that is scattered inside the cytoplasm, that's what, and it is said to be what? Endoplasmic reticulum. The endoplasmic reticulum was first noticed by Porter and Thompson. It was named ER, that is endoplasmic reticulum, by who? Porter. The name ER was given by Porter. They divide intracellular space into 2 compartments, that is luminal, that is inside the ER, extraluminar, that is cytoplasm. Let me explain this very neatly. See, if I say this is a cell, okay, just imagine this is a cell. Inside the cell nucleus is there, right? Inside the cell nucleus is there, okay, and this is the ER. Okay, this ER is dividing intracellular space in 2 compartments, that is luminal. This one, whatever the inside lumen is there, no, this thing, the brownish thing, that is the inside the ER. This is called luminal. Okay, and which is extra, which is there in outside, okay, which is in cytoplasm. Okay, that one is said to be extraluminal. Okay, this is extraluminal and extraluminal, and this is intraluminal. This is intra and this one is extra. So what I mean to say is the endoplasmic reticulum divide separately, that is divide intracellular space into two compartments. Into how many compartments? Two compartments. This is luminal one and this one is cytoplasm one. This one is cytoplasm one and this one is luminal one. This can extend from nuclear envelope to the plasma membrane. Which one? The endoplasmic reticulum can extend from the nucleus to up toward plasma membrane. Whatever I drawn, no, that one is this one. This circle is nothing but the plasma membrane. So from the nucleus, what happens? The endoplasmic reticulum is extended. Endoplasmic reticulum extends from the nucleus. Keep in mind, okay, up towards what? Towards the plasma membrane. The endoplasmic reticulum is composed of cisternae, vesicles and tubules. They are consist of what? Cisternae, vesicles and tubules. So there are two types of endoplasmic reticulum based on the presence and then absence of the ribosome. That is, first one is RER, that is rough endoplasmic reticulum, and the next one is smooth endoplasmic reticulum. If the lipids are present, if the ribosomes are present on the endoplasmic reticulum, then it is said to be what? Rough endoplasmic reticulum, because when ribosomes attach on the surface, they looks rough. So that's what it is said to be a rough endoplasmic reticulum. In simple way, if the endoplasmic reticulum doesn't have ribosome, if the ribosomes are not attached to the surface on the endoplasmic reticulum, then it is said to be what? Smooth. You can differentiate here. Let me show. See, this one is the smooth one, smooth endoplasmic reticulum, where the surface is very smooth because the ribosomes are absent. But if here these tiny round structures are there, no, they are the ribosomes which are present on the endoplasmic reticulum. This is the endoplasmic reticulum. If ribosome are present, then it is said to be a rough endoplasmic reticulum. If the ribosome is absent, then it is said to be a smooth endoplasmic reticulum. So let's talk about the rough endoplasmic reticulum first. Okay, and this rough endoplasmic reticulum organelle is said to be a protein highway because it is responsible for the synthesis of the protein. That's what it is said to be, a protein highway. Got it? Yes. So rough endoplasmic reticulum, when we go near the rough endoplasmic reticulum, you will see ribosomes attached on the surface, so that it looks what? Rough. Usually it is found near the nucleus. See, this is the nucleus. Okay, and this one is the rough endoplasmic reticulum, because the ribosomes are present. The ribosomes are attached on the surface of the endoplasmic reticulum. That's what it is said to be what? Rough. See here, there is no any kind of the ribosomes, right, attached to it. That's what it is said to be a smooth one. This is rough, this is smooth. I hope everything is clear. Next, what is the function? Functions of the rough endoplasmic reticulum. Main function is protein synthesis. Protein synthesis. Secretory and membrane proteins. They synthesize usually secretory and the membrane proteins, and the proteins transported to Golgi apparatus. Whatever the endoplasmic reticulum is synthesized the proteins, no, that protein will transported to Golgi apparatus. That's what they are said to be endomembrane system, no. Because endoplasmic reticulum, whatever it is going to produce, that is going to be hand over to the Golgi. Okay, now that's good. That's what they are coordinated. So after the synthesis of the proteins were transported to whom? Golgi apparatus. Whatever the rough endoplasmic reticulum will synthesize, it is going to be accepted by whom? From the Golgi. It is going to be transported by the vesicles, and that transported to whom? Golgi apparatus. Example, which kind of the proteins? Insulin and some of the enzymes, some of the antibodies. Okay, now these are going to be produced by the endoplasmic reticulum. Next, formation of nuclear envelope and plasma membrane formation. Even though endoplasmic reticulum plays a very important role in the formation of the nuclear envelope and then plasma membrane formation, because whatever the proteins are needed for the formation of the plasma membrane, that is going to be formed from the ribosomes which are present on the rough endoplasmic reticulum. Smooth ER formation and synthesis of the lysosomal enzyme. Whatever the lysosome, it is one of the another organelle. A lysosome is said to be a suicidal bag, and it is very important for the cell, and the lysosome has very important enzymes. Those enzymes were synthesized by what? Rough endoplasmic reticulum. These are some of the functions of what? Endoplasmic reticulum. Simple thing, just keep in one quick recap. Rough endoplasmic reticulum, that is RER, which synthesize what? Proteins. Then next it transport to whom? Golgi apparatus. Why it is said to be RER? Because it has ribosomes attached on its surface. That's it. Okay. Next, smooth endoplasmic reticulum. It is said to be, now let's talk about smooth endoplasmic reticulum. It is said to be a lipid factory. As we know, RER said to be a protein highway because it is synthesized protein. RER is said to be a protein highway because it synthesizes protein, whereas smooth endoplasmic reticulum synthesis what? Lipid. It is said to be smooth because no ribosomes on the surface. It appears smooth. Let me show you. See this one. You can see lots of difference. Imagine that you dive inside the cell and you're here. Okay. If you see, and if you go here, if you see, there will be ribosomes attached on the surface of the endoplasmic reticulum. That one is said to be what? RER, that is rough endoplasmic reticulum. If you come here, the surface looks smooth because ribosomes are absent. So it is said to be what? Smooth. So let's talk about smooth endoplasmic reticulum. And it is said to be a lipid factory because it synthesizes lipids. As we know, RER said to be a protein highway because it synthesizes protein. So SER is said to be a lipid factory because it synthesizes lipids. No ribosomes on the surface, so it appears smooth. That's what it is said to be, a smooth endoplasmic reticulum. Okay, now let me show you this one. This one is the smooth endoplasmic reticulum where the ribosomes are absent. Now let's talk about the functions. Functions of SER. First and then foremost function, lipid synthesis. It synthesizes phospholipids and steroids. Keep in mind, phospholipids and steroids. Okay, now, and it helpful for the detoxification of the drugs and poisons. The smooth endoplasmic reticulum which are present in liver cells, They are helpful for the detoxification, and they also store what? Calcium in muscle cells. They are said to be a very specialized endoplasmic reticulum, I can say, that is the sarcoplasmic reticulum. Just keep in mind, functions of the SER are lipid synthesis, detoxification, calcium storage. And rather than that, fat and steroidal hormone synthesis in animal. In animals especially, fats and steroidal hormones are synthesized by whom? SER. Just keep in mind, cell the unit of life. In this chapter, what is important is organelle, its function and its role in life of that organism. That's it. Just keep in mind what is the main role. Those all things you should be keep in mind. In NEET also, such kind of question only they are going to ask. So glycogen metabolism, glycogen metabolism is one of the function of the SER, and muscle contraction by releasing and uptake of the calcium. These all are the functions of what? SER. Main function, keep in mind, lipid synthesis, detoxification, calcium storage. What about RER? Main function, protein synthesis, and after that it is going to be transported to what? Golgi apparatus, and it forms SER and synthesize the lysosomal enzyme. That's it. Here three functions, just keep in mind. Here main function, protein synthesis you should keep, and synthesis of lysosomal enzymes. Next, some special forms of endoplasmic reticulum, just now I said, right? Those are sarcoplasmic reticulums. These are specialized smooth endoplasmic reticulum in muscle cells. They are the specialized form of smooth endoplasmic reticulum which are present in the muscle cells, especially because they stores and release what? Calcium ions, helpful in the muscle contraction. Muscle contraction, whatever the muscle contraction will be there, no, for that purpose they store and release calcium ions in where? Muscle cells. From whom? SER. That kind of SER is said to be what? Sarcoplasmic reticulum. Good. So neat line, what you should remind: sarcoplasmic reticulum regulates calcium concentration. Sarcoplasmic reticulum regulates what? Calcium concentration. So common function, if I talk about smooth and rough, what is a common function? Separately we came to know smooth endoplasmic reticulum, lipid synthesis, rough endoplasmic reticulum, protein synthesis, isn't it? Same like that, common among both will be helpful for that. Which kind of the common functions are? They provide support to the colloidal protoplasm, that is endoskeleton. Endoskeleton in the sense what? When we are going to imagine the cell will be having the skeletal support, isn't it? That means as our body is having this toughness because of our bones, right? Same like that, this colloidal protoplasm, due to the tough protoplasm, they provide support for what? Endoskeleton. They provide what? Support, because they are scattered inside the cytoplasm, right? So transport and exchange of materials from nucleus to the let me draw, okay, then you will come to know. This is cell and this is nucleus, and here it is rough, so that I am showing ribosome, and here it is smooth. Okay, transport and exchange of material. Transport, how? Whatever the nucleus, inside the nucleus is going to be produced, or else whatever the inside the ER is produced, that is going to be transported out, and whatever the outside material is there, that is going to be imported. So that's the exchange of the materials, the word is used. Then next, Localization of organelles. It keeps the cell organelles properly distributed in the relation to one another. So endoplasmic reticulum, what it keeps means it keeps the other cell organelles properly distributed. Next, formation of desmotubules, which extends through the plasmodesmata. Plasmodesmata is a part of the plant cell, okay na, it is not concerned with the animal. Here in the plasmodesmata, what happens, desmotubules were extend, they are the nothing but they are the extension of the plasmodesmata. It is very important, plasmodesmata is a very important for the plant cell interactions. You will come to know, we discussed about the plasmodesmata, isn't it? So formation of the desmotubules, which is extends through the plasmodesmata. Plasmodesmatas, from the plasmodesmata, what happens, it is going to be extended. Next organelle, which is very important and which is also a favorite topic for the NEET, that is Golgi complex. This is also one of very important organelle which comes after the endoplasmic reticulum. Golgi complex is said to be a post office or else packaging center. Why it is said to be a packaging center means, because whatever ER is going to release, no, that is endoplasmic reticulum is going to release, that is going to be transported to Golgi, and that Golgi, what it is going to do, that is going to take that material and it is going to be packaging. If say if some protein is going to be formed from the ER and then it is transported to the Golgi apparatus, Golgi is going to be pack that protein into a fine structure and it is going to be released. That's what it is said to be, what, packaging center. So Golgi complex was first named after the Camillo Golgi. Camillo Golgi discovered the Golgi apparatus, so the organelle name itself is given Golgi. Okay, na? Golgi complex is a system of flattened membrane bound sacs involved in modification, packaging and transport of proteins and lipids. Proteins and lipids. The proteins and lipids which are produced by the ER, that is going to be modified, packaged and transported. Got it? That is the main function of the Golgi. Whatever the proteins, whatever the lipids are formed by the ER, that is going to be modified, packaged and transported out. So let's talk about the structure. Made up of flattened sacs called cisternae. These are the flattened sacs. So this is said to be a cisternae. Each stack has 3 regions. Okay, each stack has what? 3 regions. First one, forming phase. This is the forming phase, that is cis phase, and this is the medial phase or else medial cisternae, and this will be the trans cisternae, maturing phase. Forming phase, maturing phase. Here it is very clear. See, this is the cis phase which is facing towards the ER. If you imagine, here it is a cell, here it is a cell, here it will be a endoplasmic reticulum. Whatever the endoplasmic reticulum is produced, that is going to be phased, that is going to be accepted by cis phase, and then it is going to be transported by trans phase. Okay, na? Formed phase, formed vesicles will be in the form of trans phase, and incoming vesicles will be towards the cis phase. Okay, next, cis phase towards the ER. Keep in mind, very important for the very important points for the NEET this session. Towards the ER, which phase will be there? Cis phase. Receive proteins and lipids. Trans phase towards the plasma membrane, release modified materials. Okay, na? Cis phase receives, trans phase dispatches. Cis is a receiver, and transphase is a dispatcher. That's it. Transport vehicles, there are vehicles which are going to be membrane bound vehicles, very small membrane bound vehicles which are going to be carrying proteins and lipids from ER to the Golgi. They are said to be what? Transport vehicles. And Golgi to the other destinations. They are like delivery guys. Okay, see here it is endoplasmic reticulum. Just imagine endoplasmic reticulum. Endoplasmic reticulum, whatever it is going to be released, that is going to be transported to transport. These are the vehicles. Okay, now these vehicles are going to give cis space. The next, after the modification, they are going to be transported to what? Whatever the transphase is going to be released, that is again it is collected with the transported vehicles. The next, whatever the other destination for a plasma membrane, nucleus, other organelles, whoever is needed, that particular kind of protein, that particular kind of secretory materials, that is going to be delivered. Okay, means from company to the customers. Okay, from company, who is going to be received, that is said to be a cis space, and that is delivered by a delivery boy. The next, after the modification, final touch up, after the modification, it is going to be released after the packaging, final fine packaging. Then next, again the transport vehicles will receive, and then finally they are going to be delivered to a particular address or else particular destination. If the plasma membrane needs, they are going to dispatch or else destinate towards the plasma membrane. See, like this. I'll show you this one later on. Now let's talk about functions of Golgi complex. The main function of Golgi apparatus is to process, package, transport and release of the secretory proteins. Processing will be there, packaging will be there, and then transport will be there. Next, they cause glycosidation of lipids. This is very important point, I can say. What they cause glycosidation of lipids and glycosylation of proteins. Please keep in glycoxidation of lipids and proteins to form glycolipid and glycoproteins respectively. Okay, now adding of the glycosylation means adding of the glucose. That's nothing but much more difficult thing. Okay, now if lipids formed from the SER is received by the Golgi, it is going to be form a glycolipid. Proteins are going to form a glycoproteins. Okay, now they are going to be modified. Acrosome of a sperm is modified Golgi apparatus. Actually, whatever the acrosome, the head part is covered by acrosome, the sperm head part, that is actually a modified Golgi. Root cap cells are rich in Golgi bodies which secrete mucilage for the lubricant of the root. Lubricant means smooth movement of the roots in the soil. That is helpful when the Golgi apparatus or else Golgi bodies are going to secrete the mucilage. Most of the proteins synthesized at ER are modified in the Golgi bodies. Most of the time. Max to max percentage of the ER, whatever the proteins and lipids are produced by the ER, that is modified Golgi body. Here question arises, why it is going to be modified? Okay, yeah, that's actually a right question. See, what actually the cell thinks, if something that is not having any kind of the information, that is not having the modifications, that is not having proper packaging on them, that cell cytoplasm or else cell other organelles thinks that they are the other materials which are not needed by the cell, so they are going to try to destroy them. That's what packaging is necessary. In simple words, I can say, if you got a delivery without package, you'll take? No, you will doubt on that, isn't it? Same like that, other cell organelles won't receive whatever directly comes from the ER. That's whatever is produced by the ER, that is going to be modified packaged, then next it is going to be transported. Same like that, whatever the companies produced in our lives, that produced material is going to be first finished neatly, then next it is modified, then next it is packaged, then next it is dispatched to our homes, isn't it? Same like that. Now, Golgi and protein modification. Next, Golgi and lipid transport. How it is going to be? Proteins from RER enters Golgi, modified by adding carbohydrate chains converted into glycoprotein. When the carbohydrate proteins is going to be added to the protein, then it is said to be a glycosylation, and that product is said to be a glycoprotein. See here, let me zoom this one. Here it is, what actually, see, this is the endoplasmic reticulum, and the protein is going to be synthesized. After the synthesis of the protein, whatever the synthesis of protein is there, no, that protein is going to be transported through a transport vehicle to the cis Golgi. Then next, from the cis Golgi, the modification will be there, and from the modification again they come out as in the form of what? Small vesicles. Then next they are going to be dispatched to the other organelles like cell membrane, nuclear membrane, wherever it is going to be, okay, like this. ER to the Golgi, who is going to be deliver transport vehicles. From Golgi to the other organelles, same transport vehicles, and that's what they are said to be, transport vesicles. Same thing, Golgi and then lipid transport. Lipid synthesized in smooth endoplasmic reticulum, modified and packed by Golgi, sent to plasma membrane or other organelles. Same thing. Here the proteins and here the lipids, because here RER is going to release protein and here lipid is going to release by the SER. Lipid is going to form a glycolipid and protein is going to form a glycoprotein. I hope this is clear to everyone. And one more formation that is important also, that is Golgi and lysosome formation. This is the NEET favorite part actually. Golgi forms primary lysosomes. Enzymes are packed into vesicles. See, just now in the last slide only I said lysosomes have very important enzymes, digestive enzymes they have, isn't it? Those digestive enzymes which are helpful for the cell, which are unwanted things are there in the cell, no, that is going to be digested by the lysosome enzymes, and that lysosome enzymes are packed into vesicles by whom? By Golgi. That's what Golgi forms what? Primary lysosome. Lysosome is nothing but a part of Golgi only. Part in the sense just the detached part, I can say. How it is going to be detached? Because it is packed. Enzymes are going to be packed in the form of vesicles. Then that vesicle is going to be turned in the form of what? Lysosome. This is what actually a main function, main important thing you should know. That is, so primary lysosome was formed by whom? Golgi. And primary lysosome has what? Enzymes. Lysosome detailed structure I am going to explain today. So this is all about what? Golgi. Let me recap. Golgi apparatus has three parts: cis Golgi, medial Golgi and trans Golgi. Cis Golgi receives whatever the receive phase is there, no, that one is cis, and then whatever the transport one is there, no, that one is trans. And if it is receives protein, it is going to form a glycoprotein. If it receives lipids from the SER, then it is going to synthesize glycolipids. I hope this is clear. Next comes about the lysosome. This is one of the important organelle, I can say. This is all about the Golgi and this is about lysosome. Lysosomes next one of the endomembrane system organelle, that is lysosome. which is said to be a suicide bag of a cell because it has a digestive enzymes. Okay, now lysosomes are single membrane bound organelle. Please keep in mind it is a single membrane bound organelle containing powerful hydrolytic enzymes. It contains what? Powerful hydrolytic enzymes capable of digesting biomolecules. They are what? Capable of digesting biomolecules. They are mainly found in animal cell. Lysosomes are mainly roughly very dominantly found in what? Animal cell. If you see the structure of the lysosome, very simple it is actually. It's surrounded by a very simple single membrane. This is the single membrane and contain acidic lumen. This is important, that is pH will be less than 5 approximately, or less than 5. Due to the acidic nature only, the hydrolytic enzymes are activated. Okay, now in the neutral nature, these enzymes are going to be inactivated, so they are going to very active inside the lysosome. When those digestive enzymes are outside the lysosome, they are just inactive. Filled with digestive enzyme, which kind of the digestive enzymes are there inside the lysosome? There is protease, lipases, nucleases, carbohydrases. These enzymes work very best in acidic conditions. See, this is the protease. This whole thing is lysosome. Very simple structure. Lysosome with a plasma membrane, single plasma membrane, and this is protease enzyme. This is lipase enzyme, and this is digestive enzyme, and this one is the nuclease. These are active only in a acidic lumen. Okay, now this is the acidic lumen. How it is going to be digesting, I will show you see inside it is going to be like this. Here, can you see? Inside, whatever the waste material is there inside the cell, no, that is going to be digested by the lysosome. That's what it is said to be what? Digestive bag also. See, this is about the lysosome. How it is going to be formed? The lysosomes are formed from what? Golgi apparatus. In the last slide only we came to know that enzymes are synthesized in rough endoplasmic reticulum. Whatever the enzymes, these enzymes, which enzymes? These one, protease, lipase, and then nuclease, carbohydrates are there, no? They are enzymes which are synthesized by whom? Rough endoplasmic reticulum, and they are packaged by Golgi and bud off as a lysosomes. Okay, neat important point. Lysosomes originate from whom? Golgi vesicles. What are the functions? These are very important. Intracellular digestion. Intracellular digestion means whatever the cell is there, if it receives pathogens, if it receives unwanted things, if there is any kind of unwanted things inside the cell, that is going to be digested by whom? By lysosome. Okay, na? Like vacuum cleaner will be there, no, in our house, same like that. Whatever the dirt will be there in our house, that is going to be cleaned by the vacuum cleaner. Same like that, the lysosome is going to be digested the unwanted things. The next, autophagy, destroy worn out organelles. That is self-eating process, it can say. That's what it is said to be a suicide bag, because whenever the organelle, any kind of the organelle is out of control inside the cell, that is going to be attacked by the lysosome, and then the lysosome himself, the lysosome itself is going to be give out or else destroy for the sake of that kind of the organelle. That's what it is said to be a suicidal bag, and that process is said to be autophagy, autolysis or else autophagy. When cell bursts, lysosomes release enzymes that is digest whole cell. This is why they are said to be what, suicidal bags. Why the main answer for why the lysosomes are said to be a suicidal bag, because when the cell is going to be burst or else some of the reasons, when cell is under the pressure, that pressure will be or else that burst cell will be released, or that cell which is having this lysosome which is inside the cell, that lysosome is releasing enzyme, and it is going to be digest the whole cell so that it can. Next comes about the defense mechanism, that is the destroyed bacteria and then viruses which are entered the cell. Think about it. We have many of the trillions of the millions of the cell, and inside that tiny cell, tiny organelles. These tiny organelles have this much of the wonders. Isn't it wonderful? Yeah, I think so. Next organelle and then last one, that is of the endomembrane system, that is vacuole. See, vacuoles are membrane bound fluid filled sacs present in cytoplasm mainly for storage and maintaining the turgidity. See, on the basis of functions and structure may differ, but on the basis of functions, you may got idea now. I think probably. See, first ER will come. What is the function of ER? It is producing protein or lipid. The produced protein is going to be received by the Golgi, and Golgi is going to be packaged, and that packaged Golgi is going to be stored in vacuole. Got it now? Why it is said to be a coordinated manner, and why comes comes about the lysosome. Lysosome is a part of Golgi only. No, that's what they are all coordinated in manner. That's what they are said to be what, endomembrane system. Now, vacuole. Vacuoles are membrane-bound fluid-filled sacs present in cytoplasm, mainly for the storage and maintain the turgidity. Turgidity in the sense watery content. Water content will be more. Especially vacuoles are very large in the plant cell. We see very small amount of the vacuole in the animal cell. Structure surrounded by a single membrane called as tonoplast. Very important. This is the vacuole. Vacuole's membrane is said to be a what? Tonoplast, filled with the sap, which is said to be a cell sap. This is not a cell. This is inside the vacuole we are now. When I go inside the cell, I found vacuole. Let me show you where I am going now. See, this is cell. I'm diving here. I came inside the cell and then saw vacuole is there. Wow, this vacuole is surrounded by a membrane. That membrane is said to be what? Tonoplast. And inside this membrane, that is a single membrane, there is again a sap which is said to be a cell sap. And this is the cytoplasm. Next, prominent in plant cell, just now I said, small and temporary in animal cell. What this cell sap actually contains? This cell sap contains water, sugar, amino acids, salts, organic acids, pigments, water materials. Sorry, waste materials. These all things will be there in the cell sap. If I zoom, you will come to know. See, water molecules, ion molecules, amino acids, many things will be there inside the cell, so that maintains the turgidity. That maintains what? Turgidity. Vacuoles in plant cell and vacuoles in animal cell. If you go for the differentiation, they are very large in plant, they are very small here. Occupies 90% of the cell volume. Just now I show you. See, this is one cell. Inside this cell, whole thing is almost whole thing is occupied by vacuole itself only. That's what. And here in the animal cell, it is only a temporary. Pushes cytoplasm towards a periphery, used for the storage and transport. Here in the plant cell, the vacuole is for the turgor pressure. Turgor pressure in the sense, the pressure which is there inside the cell. You will come to know about the turgor pressure in your upcoming classes, in your upcoming chapters like plant physiology. In plant physiology, you will come to know. Just keep in mind it maintains turgor pressure. Okay, don't be that much cocky here. Next, when it comes about the function of the vacuole, main function is storage. What it stores? It stores water, sugar, ions, pigments and waste materials. And it helpful for the maintenance of the turgor pressure, to maintain the firm and upright. And osmoregulation controls water balance. Osmoregulation means controlling the water balance. And waste disposal, stores toxic materials away from the cytoplasm. Whatever the cytoplasm will be having the toxic materials, no, that are going to be stored, and then they are keeping away from the cytoplasm, so that cytoplasm can work very firmly. Next, there are special types which is very important for the NEET, so I am going to explain this one. Contractile vacuoles, they are present in amoeba and then paramecium, which removes excess water and helps in the osmoregulation, where in the amoeba and then paramecium. This is already asked. This question is already extended NCERT when we talk. No, this question is from there. Food vacuole, temporary digestion in unicellular organism. Food vacuole is temporary, but it is helpful in the digestion in the unicellular organisms. Now, this is all about the endomembrane system. We learnt about ER, Golgi, lysosome and vacuole. Four main important endomembrane system we discussed today. From upcoming classes, I am going to start with the very important organelles, that is mitochondria, nucleus, chloroplast, other cell organelles. Okay, na? So I hope everybody is clear with the concept today. Let's start with the MCQ. First 10 questions will be for the discussion, and the last 10 questions will be for your homework. Means in the next lecture or else in the next video, you will be getting the answer for those 10 questions. Okay. First question for today's class. Dash is important site for formation of glycoprotein and glycolipids. Glycoproteins and glycolipids, where it is going to form? Site of formation, SCR, no, SCR forms lipid. Lysosome, no, lysosome have digestive enzymes. Golgi apparatus, exactly, because Golgi apparatus is the site for the formation of glycoprotein and then glycolipids. So the answer is C. Next question. The Golgi complex plays a major role. What is a major role? As energy transferring organelle? No. It is the main process of mitochondria in post-translation modification of proteins and glycosylation of lipid. Exactly, I said no, modification. Post-translation in the sense what? Nothing but after the transfer from the ER. That's the word meaning is that whatever the product is formed from the ER, no, that is going to be translated. Okay, that is going to be modified. Modification of what? Proteins and then lipids. So the answer will be of B. Exactly. And this question was asked in NEET 2013. Next, Lysosomes contain what they contains, whether they contain carbohydrates. No. What about hormones? No. What about nucleic acid? No. Hydrolases, exactly. Hydrolases are all hydrocarbons. Answer is D. Next, fourth question. How many of the following are not included in endomembrane system? Today we learnt about endomembrane system only. Endomembrane system has what? ER, that is endoplasmic reticulum, yes. Next, Golgi complex, yes. Lysosome, yes. Mitochondria, no. Chloroplast, no. Vacuole, yes. Peroxisome, no. So among these 7 we have 1, 2, 3, 4 endomembrane systems, isn't it? So the answer is fourth one. Oh, sorry, it's my mistake. The answer is 4, not B. It's a C. Answer is C. Sorry for that. Next, fifth question. Which side in a cell does luminal and extraluminal compartment are situated respectively? Which inside or which side of a cell does luminal and extraluminal? Extraluminal will be outside the ER and then luminal will be inside the ER, right? So luminal will be cytoplasm and inside the ER respectively, he is asking, right? Which side in a cell does luminal? In a cell, luminal will be inside the ER and then extraluminal will be in the cytoplasm. So the answer will be B. Yes. Next, which of the following statement is not correct? Not correct. The hydrolytic enzyme of lysosomes are active under acidic condition. Correct. This one is correct. Lysosomes are membrane bound structure. Exactly. Lysosomes are formed by the process of packaging in a endoplasmic reticulum. No. Lysosomes have numerous hydrolytic enzymes. Correct. So this is already asked in NEET 2019. So what is the correct answer? The right answer is C, because it is wrong. Seventh one. Hydrolytic enzymes are activated at the pH of acidic. First answer itself is right. In the neutral, they are going to be inactivated. So hydrolytic enzymes, why they are present inside the lysosome? Because lysosome has a acidic condition. So the answer is acidic. Eighth question. Water soluble pigments found in plants cell vacuole are carbohydrates, anthocyanins, xanthophylls and chlorophylls. They are the anthocyanins. This one I asked because you should know cell vacuoles of plant has anthocyanin, which is already asked in NEET 2016 paper 1. Okay. Ninth one. Last two questions I have arranged this time assertion and reasoning type of questions. You people already know about this. They are going to give you 4 options like this. Which one is correct? You should read the 2 statements. The next, you should compare which one is the correct option among the below. Okay, let me start. Assertion. S in 80s S represent sedimentation coefficient. We are talking about the ribosomes here. Sedimentation coefficient is direct measure of density and size. S in ATS, S alphabet in ATS represents sedimentation coefficient. It is correct, but that won't measure the size of the ribosome. So the answer is assertion is true, but reason is false. The last question, that is assertion. What assertion is there? Lysosome possesses acidic pH. Of course it is possessing acidic pH. Lysosomes are rich in hydrolytic enzymes which are activated by acidic pH. So the answer will be, if both assertion and reasons are true and the reason is correct explanation of the assertion. So this is the correct option. A is correct for the 10th question. Now my work is over. Now your work starts. Take your pen and paper, write these questions and try to answer them. They are very interesting, isn't it? Membrane bound organelles are absent in, it is already asked in AIPMT in 2010. Options here are Saccharomyces, Streptococcus, Chlamydomonas and Plasmodium. Answer yourself. What is a tonoplast? Outer membrane of mitochondria, inner membrane of chloroplast, membrane boundary of vacuole of plant cell, cell membrane of plant cell. Next, match the column 1 with column 2. Next question. Peptide synthesis inside a cell takes place in mitochondria, chromoplast, ribosomes and chloroplast. Peptides here is nothing but the units of protein. So protein synthesis takes place by whom among them? You should know that. Which of the following are not membrane bound? The Golgi complex participate in. Which of the following organelles provides cytoskeletal support to a cell? This is already asked in JIPMER 1998. Better answer your answers in comment section. Which of the following is correct about the vacuole? Among this, which one is correct about the vacuole? This one. The name endomembrane system to the membranous organelle because it is asked question in 2021. Which one of the following cell organelle is enclosed by a single membrane? NEET 2016. So these all are the 10 questions which is homework for you. Prepare your answers so that you can check your answers in upcoming class. So from the last class, whatever I asked to do, that homework answer is this one. You may check your answers. So last but not the least, today's motivational thought, that is motivation through the microscope. We take example as a Golgi apparatus only. You know already Golgi apparatus is for packaging and processing. So take 10 minutes of short break, step aside or else step away from your desk, let your brain process the informations, grab water, snacks to replenish your glucose level to the brain. So rule what you should maintain is no social media. Don't clutter the vesicles of your brain. Use your social media for your useful purpose. Don't waste too much time on that. Okay, thank you. Welcome for the next session. Bye bye.